All right, guys, welcome to Chapter 11, Section 1, where we are going to talk about basic patterns of human inheritance. Here's a fact stated for you right here. The inheritance of a trait over several generations can be shown in a pedigree. Okay, so I'm going to take a second to do what we always do in class on these kind of notes, and you might have to pause it. So, first, I want you guys to think about something you already know about inheritance and traits being passed on. Think about something you might learn about in this lesson, something you might want to learn about, or you think you might learn about, and then later you can think about something you learned. So take a second to pause and think about that. Okay, let's keep moving. Here are our three essential questions, or our three main ideas about today's lesson. How can genetic patterns be analyzed to determine dominant or recessive inheritance patterns? What are examples of dominant and recessive disorders? And how can human pedigrees be constructed from genetic information? So if you can remember those three questions, the answer to those three questions when we are finished, you are in a great spot. Okay, first let's look at recessive genetic disorders. A recessive trait is expressed when the individual is homozygous recessive for that trait. Now let's not forget that homozygous recessive, that would be in your Punnett square, two lowercase letters. Those with at least one dominant allele will not express the recessive disorder. So it could be two capital letters or even just one capital letter with a small letter in your Punnett square for the genotype, and it is not going to express the recessive disorder. An individual who is heterozygous for a recessive disorder is called a carrier. So if they are heterozygous, meaning big letter, little letter in their genotype, they have that gene, but they're only a carrier of it. They do not express it. Okay, cystic fibrosis. This is a disorder that affects the mucus-producing glands, digestive enzymes, and sweat glands. Chloride ions are not properly transported out of cells of a person with cystic fibrosis. And cystic fibrosis causes mucus excretion that clogs ducts in the pancreas, interrupts digestion, and blocks respiratory pathways in the lungs. This is a pretty bad recessive genetic disorder to have. We also have albinism. Albinism is caused by altered genes resulting in the absence of skin pigment, the skin pigment called melanin in hair and eyes. Individuals with albinism have very pale skin, white hair, and pink irises. So no matter what the person's ethnicity or race or where they come from, none of that matters. If you have albinism, your skin is white, like the color of milk or paper. Okay, we have Tay-Sachs disease. This is caused by the absence of the enzymes responsible for breaking down fatty acids called gangliosides. And gangliosides accumulate in the brain, inflating brain nerve cells and causing mental deterioration. This one's bad because it affects your brain. That's not good at all. We have galactosemia. This is a recessive genetic disorder characterized by the inability of the body to digest galactose. So galactose is a milk product, and when you have galactosemia, you cannot digest milk products. Now here are some examples of dominant genetic disorders. If the disorders are dominant, that means the genotype is going to be two capital letters or a capital letter and a lowercase letter. Huntington's disease is one of these. It affects the nervous system. It causes gradual loss of brain functions. It occurs in about 1 out of every 10,000 people in the United States. Achondroplasia, this causes small body size and limbs that are comparatively short, and it's caused by an abnormal gene that affects bone growth. So it makes you have really tiny arms and legs. Okay, when we talk about these things, it's important to know what a pedigree is. A pedigree is a diagram that traces the inheritance of a particular trait through several generations. This picture here that you can see on your screen is an example of a pedigree. On the left it gives you kind of a key for some of the symbols here. The yellow circle would be a normal female. The kind of reddish circle would be a female who expresses the trait being studied. And then the one that's kind of half and half, yellow and red, a female who is a carrier for the trait. She doesn't express it, but she has the gene. When you see a vertical line, that represents you're moving on to another generation. And just like these here, when you see horizontal lines between two individuals, that means they are parents together. And then when you see the lines that kind of go together like this, those are all siblings together. Okay, the yellow square is a normal male, a dark reddish square is a male who expresses the trait being studied, and then a half and half yellow and red square is a male who is a carrier for the trait, but he does not express it. The Roman numerals represent each generation, and then Arabic numerals, meaning just regular numbers like 1, 2, 3, 4, those are individuals in that generation. So generation 1 has 2 individuals. Generation 2, we see 4 individuals. We see a normal male who had children with a female who's a carrier for the trait but does not express it. And their offspring, they have a normal female, they have a male who's a carrier for the trait, a normal male, and a female who is a carrier for the trait. So it's a 50-50 chance here to have that trait in this pedigree. Now, as we analyze pedigrees, pedigrees can be used to examine both recessive and dominant genetic disorders. They show information about an individual's genotype, and they can be inferred from the phenotype of his or her parents and the offspring. So they can look at this pedigree here on top of a recessive disorder, and they can see that these two parents here are carriers for, let's say, Tay-Sachs disease, and in their offspring, the odds of their children having this disease came out to be 3 children without it and 1 child that completely has that disorder. Like, it has 2 recessive genes that comes together to make it express that trait. Now, the dominant disorder, we have 1 parent without, 1 parent with, and you can kind of see their offspring. It looks like they had 7, let's see, 1, 2, 3, 4, 5 offspring. 2 of those offspring came out with a disorder, 3 did not. And then you can see they married someone or had a child with someone. They did not have the disorder, and over here, they did not have the disorder as well. Okay, as we analyze pedigrees, we can infer the genotypes. Knowing physical traits, which would be the phenotypes, can help determine what genes the individual most likely has. So again, knowing what the traits they have physically can help you kind of take a guess or infer what genes they actually have. And then when it comes to predicting disorders, record keeping helps scientists use pedigree analysis to study inheritance patterns, to determine the phenotypes, and then to ascertain or eventually figure out the genotypes. Okay, that is chapter 11, section 1. If you need to go back and read the textbook a little bit to get a few more details, go for it. If you need to ask me any questions to clarify something, send me an email or a Google Hangouts chat or whatever works for you. And now find your way to this week's discussion board.